All right. So this is a continuation of last lesson. So we're going to take a kind of a deeper look into it. There are a lot of things. You guys should know most of the things I'll talk about today. We did them either Friday, last class, or we did them in the first semester. So let's see what you can actually remember. So first off, what type of organism or what type of cells DNA is easier to regulate and express? Prokaryotic or eukaryotic? What's easier? Prokaryotic is easier. Why is prokaryotic easier? You can't just say what it is. You have to know why it is. What is it about prokaryotic DNA that is easier to regulate and express?
Because they're all at once. Like it like has operons and it can do all of it at once.
Okay, it has operons. It can do multiple things at one time. That is correct. I'll add that in a minute. What about the quantity of DNA? Is there a lot more DNA in a prokaryote than us, the eukaryotes, or is it the opposite? So there's much less DNA. Much less DNA than eukaryotes. In fact, do you remember how many chromosomes a prokaryote has? How many? It's between 0 and 2. 1. Know that. They have 1 chromosome. I mean, that's easy. How many chromosomes do we have just as humans? 46. Also, as Emmy said, they can transcribe and translate simultaneously. It's like reading a book, or excuse me, it's like writing a book and reading it at the same time. So they can transcribe and translate simultaneously. Us eukaryotes can't do that. Not because we're like inefficient. It's just that we have a lot of information. We have much more DNA than a prokaryote. In fact, I told you guys it's last week. Prokaryotic DNA is like a 1-year-old toddler's book. Cow says moo. Sheep goes bah. Fox goes da-da-da-da-da-da. While as a eukaryote, it's like trying to read the entire encyclopedia. It's just a lot of information. So I'm just going to say they have more than 1 chromosome. Depending on what type of organism you are, it varies. Dogs have 78 chromosomes. Humans have 46. I think chickens have like 72 or 78. Let me ask this. Hey Siri, how many chromosomes does a lion have?
Lion have. Check it out.
It doesn't tell me. Hey Siri, how many chromosomes does an African elephant have?
Okay, I found this on the web for how many chromosomes does an African elephant have. Check it out.
Oh, there's 3 species. Sheesh. Anyway, point I'm making is a lot of different species have lots of different numbers of chromosomes, but when it comes to prokaryotes, it's pretty much 1. And because eukaryotes have so much information, we cannot transcribe and translate simultaneously. We cannot walk and chew gum. We can only do one or the other. Okay, so we are going to go over those 5 stages of eukaryotic regulation again, but I'm going to go a little bit deeper into it. And as I said, you should know more than half of what I'm talking about. So there will be some new stuff. But can any of you tell me what is that first stage of eukaryotic gene regulation called? Look at your notes on Friday if you need to. Chromatin structure. This takes place in the nucleus. Don't forget that. Now, There's two types of chromatin, and these are two new words you learned on Friday. What is the chromatin that is densely packed called, that cannot be accessed by RNA? Heterochromatin, nice. So there's a few characteristics of heterochromatin we need to be sure you all know. First one, it is densely packed. What made it so densely packed? What chemical group does it have with it? What functional group? It's been what? It's been methylated. A methyl group is a CH3 group. This type of chromatin is inaccessible by RNA polymerase. If it's inaccessible by RNA polymerase, can it go through transcription? So there will be no transcription happening today. If there's no transcription, then there's no translation, then there's no protein, just chain reaction. Okay, now what do we call the chromatin that is not densely packed? It's loosely packed, and it can be accessed by polymerase. Euchromatin. It is loosely packed. That's because it is acetylated. An acetyl group is a C2H3O2 group. And because it is so loose, it can be accessed by RNA polymerase. So I'm going to give a green light to transcription. You can transcribe, which means that there is hope for translating. Any questions, comments so far? Nothing so far is new. We haven't done anything new yet. Okay, this next part is not new either. I want to give an example of heterochromatin, and we learned this back in Unit 5. What do you call a cat that is orange and black? Calico. Now, what gender of cat can be calico color? Why is that? Well, ladies, how many X chromosomes do you have? And let's see if you know this, because like I told you, teachers have sixth senses they have a sixth sense on whether their students get something or not, just by the body language and the vibe. Where are your sex chromosomes located? Where, Emmy? Which ones? Thank you. Good. Hope you all were thinking the same thing. Your sex chromosomes aren't every cell. Some people think, I thought they were only in my sex organs. No. I thought they were only in my ovaries. No. They're everywhere. They're in your fingers. They're in your head. They're in your shoulder. They're in your organs. They're in your legs. They're in your feet. They're in every cell in your body. Every cell. Now, ladies, you guys, or girls, you have two sex chromosomes. X, X. They kind of look like this. This should be nothing new. Fellas, how many sex chromosomes do you have? What are they? Where are they located? Anybody? Everywhere. Good, just like the girls. However, look at the size difference here. The Y chromosome is embarrassing. It doesn't really do much. It has the SRY gene, which is really what makes a male a male. But other than that, it might have a little trait here or there. But the X chromosome has just so much more information, even information that's nothing to do with your gender. But here's where the problem lies. Girls, you have 2 X chromosomes. Guys have 1. Not fair. So girls, in every single one of your cells, what happens to one of your 2 X chromosomes? It gets methylated. That's right. It's because males and females are supposed to be compatible. Now, when I say it's methylated, does that mean it disappeared? No, it's just covered up. It becomes inactive now, which means it cannot be expressed. This is a bar body. Girls, when you are going through meiosis and you're producing your egg cells, it gets turned back on so that you will pass on a fully functioning X chromosome to your future son or daughter. But as it is right now, one of your X chromosomes is turned off. But when your cells go through meiosis to produce your eggs, it gets turned back on. So you don't have to worry about passing on a shut down chromosome to your kids. Now, when it comes to that cat, let's say that a B allele makes the cat have black fur and an O allele makes the cat have orange fur. Cat fur color is a sex-linked trait. So here is a female black cat, here is a female orange cat, and here is a female calico cat. Now, which of the X chromosomes is actually going to be the bar body? I don't know. Flip a coin. You really can't predict that. Maybe one day we can learn to control it. But this drawing should look familiar. I've done this before with you guys. If I have a cat that has this part of its face orange, which chromosome is the bar body? The X chromosome with the black gene or the X chromosome with the orange gene? Right, so everything here is XB, XO, but definitely the one that's carrying the black gene is methylated. So it's basically covered up. It's shut down. And then vice versa. Everything here, XB, XO. The one that has the orange allele is methylated. They are still there, they're just shut down. That's how the calico cats do it. And that's an example of the bar bodies. One more example when it comes to chromatin structure, that's going to be epigenetic inheritance. In epigenetic inheritance, the pattern of inheritance doesn't depend on the genes. So that can I remember learning that for the first time. I'm like, what? I didn't get it. So here's what this means. We've gone over this several times. This will probably be the last time before we start getting ready for your AP exam. So take this in. Anybody, what is that genotype called? Big A, little a. Heterozygous. Now, if I told you that the dominant A is brown hair and the recessive A is blonde hair, just by looking at the genotype, what would you expect the phenotype to be? Yeah, you would say brown. And you would have no reason to be wrong. Well, let's say I open door number 1. Here's the person with a heterozygous genotype, and they're blonde. Wait a minute. Is their hair dyed? No, they're natural blonde. How in the world is that? Which allele is methylated? The dominant one. And so what is the only other alternative for this individual? Blonde. That's the only one that's left. That's right. And so this is an example of epigenetics where this person is heterozygous and you have no reason not to think that they're not going to follow that pattern. But this is an example of a form of inheritance that is not dictated by the genes. The last thing I want to go over here, and this is something new, is going to be called genomic imprinting. Mom or dad pass on methylated alleles to offspring. This occurs during gamete formation. So while the sperm and the egg are coming together, they got methylated. Now, sometimes this can happen naturally, but I want you all to hear me now. You all are 16, 17, 18. One day you may want to have a family. Some of you are going to get pregnant. Some of you are going to have kids. Or some of you are going to have wives that are going to have kids. A lot of the behaviors, I shouldn't say a lot, some of the behaviors that you may partake in can be considered risky behaviors. Smoking, being around people who smoke, drinking, eating or ingesting some sort of toxins. That can actually affect the development of your offspring, and that could affect the development of your own gametes, your sperm or your egg. So let's just say that we have mom of the year here. She's pregnant, and I just drew a really big abdomen on her. So she's pregnant. We're going to call her first generation. And by the way, she's smoking, so that's a little cigarette. Here's some smoke. Here is the baby, and if you didn't know anything about babies, they're in the 3rd trimester, they're upside down, and they're backwards. So this kid is upside down. This baby here is 2nd generation. And let's say this is a baby girl. She already has all the eggs she's going to have her whole life in utero. All you girls got all the eggs you're ever going to have while you're still in your mom. That's 3rd generation. So if this mother is smoking, she could imprint onto her, not only her baby, but her potential grandchildren, traits that are going to be methylated without them really even standing a chance, because the mother partook in risky behaviors. Things like smoking, secondhand smoke, other stuff, drugs. So it's pretty important that you really try to live as clean or healthy of a lifestyle, especially while you're pregnant, as you can, so that you don't run the risk of having any issues. Now, can you play by the book and do everything right and still happen? Yeah. There's people that have never gone near a cigarette and they get lung cancer. It happens, but don't use that as an excuse to justify a bad habit. And if you want to talk about vaping, guys, we don't know what it does to the body entirely yet. So much less do we know its effects on reproduction and pregnancy. So unless you want to be the test guinea pig, steer clear of that too. It's still a very new thing and we don't know what it does. But anyway, that's genomic imprinting. You could actually damage the alleles before the child is even conceived yet. All right, let's move on to transcriptional control. Number 2. I'm going to try to do this one really quick because I ran out of time last period. Now, guys, red are exons, blue are introns. Which ones are in, let me rephrase that. Before I get to that, know that in transcriptional control, you start with DNA, you end with pre-mRNA. The reason that this is called pre-mRNA is that you have exons and introns. Do you remember which one needs to be gotten rid of? The introns need to be gotten rid of. And I am pretty sure I did get to this with this class, but even if I didn't, or even if I did, I want to add to it. I want to show you a few examples from the AP Biology exam that at least are on the mock exams I give every year. And let's see how you stack up. So if I think I did get to this question with this period. If I didn't, then I didn't. But here we go. You don't have to write this. I'd rather you guys just watch and take this in. So if you want to give yourself a little breather, that's okay. Okay, so we got A's, U's, C's, and G's. Let's see if I can lower that. Okay, cool. Now, let's try to make this a little easy here. Put that there, 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 there. Perfect. Okay, first question. Tomorrow, we're going to do mutation. I think you're really going to enjoy tomorrow's lecture. I know today's kind of boring. I get it. I know. But tomorrow should be pretty entertaining because it's mutation. Well, one of the types of mutations I'm going to teach you is called a substitution mutation, which is exactly what it sounds like. You substitute one base for another. So, Ben, let's say that this A right here, I erase that A and replace it with a C. Is that going to affect, given its location, that's a big factor here, is that going to affect the protein that will eventually be made from the mRNA? The answer is no, and the answer is correct. So why not? Yeah, your justification is fine. It's an intron. It's not going to be part of your final mRNA. That's absolutely right. I have nothing more to add. Next problem. Let's make that A come back. Trey, how about this? Trey, when you're standing in line at lunch, let's say someone in front of you just leaves the line. What does everybody behind that person naturally do? Just kind of move forward. That's called a frame shift, guys. So, Trey, what would happen if I deleted this A? It's gone. That's called a deletion. And then everything moved up one and filled that void. Is that going to affect the protein? Yeah. Yeah. Because, guys, this G right there, that was on an exon. You were supposed to keep that. But because it's now an intron, you're going to cut it out. That's not good. And so that will affect your protein. Now, on the other hand, Trey, let's say that you have the other type of, the opposite of a deletion is called an insertion, or you can call it an addition. You squeeze another A in here. So watch this, Trey. I move everything over one, and I squeeze in another letter. Is that going to affect the protein? Yeah. Yeah. This A right there was part of the intron. That's supposed to go. But now you're keeping it because it's on an exon. So I want to share with you what your friends that were in this class last year did. As soon as they see a mutation take place in the intron, they're like, oh, it's not going to matter. When it does, you have to just take it on a case-by-case basis. So I can tell you that when you're taking your exam, you're going to remember little things like this. Oh, it was on the test, Mr. Thorson, it was on the test. I'm like, good. Did you know it? Yeah, great. All right, two more, and then we'll move on. Zoe, what if you accidentally delete an exon? Oops. Is that going to affect your protein? Yeah, I mean, very much. Lauren, what if you delete one intron, like you should, but you don't delete the other one, and then you splice it all together? So you have an intron part of it when it shouldn't be. Is that going to affect your protein? Yes. So you should delete the introns, you should keep the exons, but if you accidentally keep an intron or if you accidentally delete an exon, you can bet that will affect your protein. All right, that's really nothing else I have to add when it comes to transcriptional control. Just know that part. DNA to pre-mRNA. Let's get to number 3. Post-transcriptional control. This is where you're going from pre-mRNA to final mRNA. What is the difference between what does final mRNA have that the pre-mRNA doesn't? I can think of 4 things. Anybody want to take a shot at it? What does final mRNA have that pre-mRNA doesn't? Okay, so I'm going to rank them here. That's actually the last one we'll do. It has a 3' poly A tail as an adenine. That protects the mRNA in the cytoplasm. The cytoplasm can be a very scary place for mRNA, so it needs some protection. It's like a baby bird out of the nest. What else? All right, it has a 5' cap, which is mostly guanine. That aids the mRNA in exiting the nucleus. Okay, what else? What happens in the introns? Introns are removed. What happens after the introns are removed? What do you do with the exons? You splice them together. Splice the exons. That's the 4 major things that separate pre-mRNA from final mRNA. All right. That's it for part 3. Let's keep on going. We have 9 minutes. Okay, translational control. You have final mRNA, and that's going to result in a non-functional protein. Now, there will be some new information here. The length of A, the 5' cap, B, the 3' poly A tail, and C, the length of final mRNA will affect how long translation is and how long the protein is. And now I have something entirely new for you. I'll let you all get caught up. Here's your brand new concept, miRNA. This is microRNA. I'll tell you about this stuff. Here we go. They regulate translation by destroying mRNA. When this happens, translation doesn't even happen. MicroRNA comes from pre-mRNA. Here's the craziest part. MicroRNA is complementary to mRNA. So if you don't know what I mean by that, here's what I mean. If this strand of nucleic acid is mRNA, and it says AUGCA, microRNA will complement it. UACGU. Here's why this is important. This forms a double helix RNA. We've never mentioned that before. A double helixed RNA. DNA is supposed to be double helix, not RNA. Well, here it is. Here's the issue. This is how it's a problem with translation. Double helixed RNA can't be translated. So what's the purpose? It regulates translation. Not everybody, not everything that DNA codes for can be made. Sometimes you have to say no. And so there are genes that just will not be expressed. That's natural selection. All right, and we have 4 minutes and we are going to finish, thankfully. This last part is just a little bit. Post-translational control. Did I write? Hold on. I didn't write. Oh, I did. Okay. Final mRNA in a non-functional. So this last part is going to be non-functional protein to functional protein. So basically, proteins that are no longer viable must be destroyed. And this is done by proteosomes. Proteosomes will destroy proteins that have worn out their welcome. They're done. You don't need them anymore. And that is it. So guys, if you could just listen to me before you start packing up. This is one of those lessons where I know it's not that interesting. It's not that riveting. I get that. But this is one of the lessons where you have to learn the finer details to really understand the bigger picture. So be sure that you actually do yourself the service of studying this over time. You have a test next Wednesday. You should start studying now. You cannot eat an elephant in one bite. You have a lot to go over. And we still have more stuff coming. So take 20, 30 minutes to look over this material. Most of it was not new. Most of it was stuff from last week. Definitely focus on the new stuff. Be sure you have it understood. And tomorrow should be fun. We will do mutation. Mutations are typically pretty interesting. All right, that's it.